Hi everyone, my name is Abra Vasquez and my project explores how body size might influence ovarian follicle density across mammalian species. The ovary is a complex organ made up of many tissue and cell types. Within it, ovarian follicles serve as the fundamental unit of a female fertility, each follicle containing an oocyte surrounded by supporting cells that regulate its growth and development. What makes follicle density interesting is that it reflects a species' reproductive investment and life history strategy. Smaller mammals often reproduce more frequently and have higher follicle turnover, whereas larger mammals produce fewer follicles and invest more resources into each offspring. Traditionally, ovarian structure is studied using two-dimensional histological sections stained with hematoxylin and eosin, or H&E. However, newer approaches like MRI, CT, and tissue clearing now allow for 3D reconstruction of ovarian architecture. The figure here from Zhang et al. 2021 shows how follicles progress from primordial to Graafian stages and highlights some of the signaling pathways involved in follicle maturation. It gives a good overview of what I'll be analyzing in this study. The main question I'm asking is there a relationship between mammalian body size and ovarian follicle density across species? My hypothesis is that smaller mammals will have higher follicle densities than larger mammals, reflecting their faster reproductive turnover and shorter life histories. To explore this, I'll use images from the Mother database, which provides standardized ovary histology for multiple species. By comparing follicle density and body size, I aim to observe general scaling patterns that could indicate a link between reproductive strategy and morphology. For this project, my approach includes 5 main steps. First, I'll select species from the Mother database that have high-quality ovarian sections and complete metadata. Second, I'll quantify follicle counts per unit area using visible follicles and H&E stained images. Third, I'll gather published body mass data for each of those species. Fourth, I'll compare follicle density and body size across species to see if a pattern emerges. And finally, I'll summarize the observed patterns to highlight general trends. The Mother, or Multi-Species Ovary Tissue Histology Electronic Repository, is the primary data source for this project. The database contains standardized ovary images from a variety of species, each with metadata including donor age, section, thickness, and staining protocol. I chose 3 examples to represent variation in size and reproductive pace. First, the house mouse, an adult specimen showing high follicle turnover typical of small mammals. Second, the Japanese macaque, a larger primate with slower reproductive pace and a lower follicle density. And third, the fathead minnow, which I included to showcase the diversity of the Mother dataset, though it won't be part of the mammalian comparison. Together, these examples highlight the breadth of Mother's dataset and provide a foundation for cross-species analysis. This exploratory study helps identify a potential biological patterns connecting body size and ovarian follicle organization. It also demonstrates how existing histological data can be repurposed for comparative research, reducing the need for new sample collection and promoting data reuse. Future directions could include expanding the dataset to include more species, refining follicle counting with automated image analysis, or exploring how follicle size relates to age and reproductive phase. Overall, this project provides a small but meaningful step towards understanding how morphology and life history traits interact in mammalian reproduction. Here are my references. Thank you for listening. I'd be happy to answer any questions about my presentation under the comments. Thank you.